So we are recording. Like I said, I'm going to mute you, all of you, and just remember to hit your space bar if you want to talk or use the little chat feature. All right, now I'm going to share my screen and share my full desktop. Oh, but of course now when I share my desktop, my little chat feature goes away. Let's see if there's a way to bring that back. And okay, there we go. All right, so now I can see the chat if anybody wants to use that option. All right, so hopefully everybody can see my screen at this point. And you'll see that I'm logged into my courses. And in the content for this week, the very last entry here is a link to a kind of crude web page that I put together, which is a list of databases for locating molecular models. There's also a few other things on there, like links to some of the cell image databases and the electron microscopy databases that we talked about earlier in the semester. So let me click on this and open it up. And here you see the list. And the links to the molecule databases are organized into two groups. First, there's a group of databases here for locating what are called small molecules. So these would be some of the smaller biological molecules, like the individual phospholipids in the lipid bilayer, or things like ATP, vitamins, cholesterol, hormones, all things that are generally considered relatively small. Then there's another set of links to what are called macromolecules. These are the really big ones. This primarily refers to things like proteins, which are very large molecules. There's also at least one nucleic acid database in here for accessing structures of DNA and RNA and other things that are based on nucleotides. And then there are a few other things that I'll show you shortly. So it's important when you're searching for a molecule to, first of all, do a little bit of research about that molecule to know what it is you're even looking for. Is it one of the smaller molecules, one of the smaller biological molecules, or a drug or something like that? Or is it one of these macromolecules like a protein or nucleic acid? Because you would probably end up using very different databases to search for those different kinds of models. So let me scroll back up here to the small molecule databases, and I'm going to show you 3 of these. There are others here, but I don't want to take the time to walk through every one of them. Between the 3 that I show you, you'll probably be able to find models of almost any small molecule that you could ever want. The first one I'm going to show you is PubChem. This is a US government database, and as you can see here, it contains over 94 million different compounds. So it's pretty hard to not find something that you're looking for in here. When I click on that, it brings us to this page, and there's just a simple search feature here. I'm going to look up, let's look up ATP. I could probably just type in ATP, but I'll probably have better luck if I type out the full name, adenosine triphosphate, and then you can just hit return. And it will bring you to a list of quite a few different structures. But if you look at the names of some of them, you'll see that some of them are kind of odd, like manganese ATP or epsilon ATP. I don't even know what that is. But typically, the first one that pops up here is the sort of standard generic ATP molecule. So you can click anywhere on here on the image or on the name to open up a new window with some more information about it. There's a little tiny thumbnail of its structure. There's the chemical formula and some alternative names for it, some synonymous names. And then there's a description of the molecule. You know, what is it and what does it do? And oftentimes that's a great source of information to learn more about a particular molecule that you're illustrating. If you continue to scroll down, there are a couple of different options for structures. This is item number 1.0, and then there's 1.1, which is two-dimensional structures, and 1.2 is called 3D conformers. These would be 3D models. You want to be careful not to download the 2D structures. It is possible to download a structure from this 2D section, but it's going to give you a molecule that's completely flat. It won't have any three-dimensional, you know, dimensionality to it. So you want to make sure to scroll down here to where it says 1.2 3D conformer. There is a little 3D thumbnail of that you can scroll around. But what we want to do here is just go to the downloads menu here under 3D conformer. There are 4 different file formats that you can download this in. The only one that's going to work pretty consistently with the molecular visualization software that we've downloaded, things like PyMol and Chimera, the only one that's going to usually work with those is this first one called SDF. So I'm going to click to save an SDF file. It's going to save it directly to my downloads folder. I've got my downloads folder open, and you can see here it's Conformer 3D CID 5957. That 5957 is just PubChem's code number for this particular molecule. I mean, if you want, just to make sure you don't forget about it, just rename the file, you know, in this case, atp.sdf. And if I double click that, at least because I've got Chimera set up as my default program for molecular models, it opens up the ATP model. Just, you know, to give you a little chemistry lesson while we're in here, the left-hand part of this molecule, the 2 rings with the blue and tan, that is adenine, the, you know, one of the bases, of course, that's found in DNA and RNA. The blues are nitrogens, the tans are carbons. That double ring of adenine is then connected to this other ring down here in the bottom left. That's a sugar. That is a molecule of ribose, one of the sugars that's also found in, that would be found in RNA. And then you see there are 3 orange atoms over here on the right with a bunch of oxygens connecting them together. These 3 orange atoms, maybe it'd be easier if I made it ball and stick. The 3 orange atoms are phosphorus. So you've got adenine bound to a sugar that makes up what's called adenosine. And then you add 3 phosphate groups to that and you get adenosine triphosphate. Close that session and then go back here. So that's really all you need to know about PubChem. Very easy to search, lots of information about your molecules, and just make sure that you use the 3D conformer if you want to download an actual 3D model. So let me go back to my list of databases. I'll show you the next one.
Which file do you save from the 3D form?
The SDF.
Thank you.
Yep. So the next database is another one that's hosted by the US government, this one particularly from the National Library of Medicine. This one has a lot fewer structures in it. Last time I updated this webpage, it was about 200,000. It may be more than that now, but it's a far cry from the 94 million on the other one. But still, it's a good resource to check out. So ChemID Plus, there are a variety of ways that you can search for molecules. You can actually search for them based on whether they're toxic or not, or certain physical properties like melting point and things like that. But you know, we're not going to use those. We're just going to use a simple name search. So you just enter something here in this blank space up at the top. This time I'm going to pick sphingomyelin, which is a phospholipid that I've shown you before. You can just hit that and then hit return on the keyboard, and there's only one entry here for sphingomyelin in the ChemID+. There's more information here about it, although it doesn't really give you any description of the molecule like you'd get with PubChem. In order to access a 3D model of whatever molecule you're searching for here using ChemID+, you'll see there's a little two-dimensional cartoon of the molecule, and then there's a little button that says 3D. When we click on this, it's going to open one of these web-based applets to allow us to view this thing as a 3D model. So it opens up a little program called JSMole, and it's automatically spinning around. We can just click this little button here to stop the rotation. You can actually change some of the representation of the molecule here between ball and stick, space filling, wireframe. But the more important thing here is that you're able to click right on the molecule itself, right in the image window, and you can access a whole bunch of other menus to do things to it. This is something where you actually have to right-click right on it, and I did actually go into my preferences on my computer and change the preferences from my mouse so that I actually have a right-click function. Usually with the Mac, if you hold down Control key and left-click, it's supposed to act like a right-clicking, but that doesn't happen to work with this particular application. So I had to actually change my mouse so that it does have a right-click function. So here I've right-clicked on the molecule, and you can see we get a whole bunch of menus where we can, you know, change the scale and add surfaces and styles and so forth. But the only thing I'm interested in right now is this very first menu, File, Save, and then it's a little annoying because the text kind of overlaps itself. But the very first entry here under Save is save a copy of this particular molecule, and it gives a code number. It's just giving me the code number here, but I'm going to click OK. And it's asking me what I want to do with it, and I'm just going to say save the file. I'm going to go back to my downloads folder, and we'll see that it did save the file here. This 85187. Now, if I double click on that, I know what's going to happen. It's just going to open up a text editor, because currently this is just a raw text file. It's opened up this text editor on my Mac that shows me what the file looks like if I explore it, but I don't really want that. I'm going to quit the text editor. What I've discovered is that there's a little trick to fool the computer into recognizing this as an actual three-dimensional molecular model. I have to click on the name of the file to highlight it, and then go to the very end of the file name and add a molecular model format suffix or file type. And I found that if I rename it as a mol file, MOL, that then it'll open in Chimera or other molecular modeling programs. So I've simply added the .mol file type to the end. It's going to ask me, do you really want to do that? And I'm going to say, yes, I do. Now, when I double click it, it opens it up in Chimera. So this is sphingomyelin, one of the common phospholipids in the lipid bilayer. So it's a little bit awkward using this ChemID Plus, just because, like I say, you have to right-click the molecule, save it. What you're saving is just a plain text file, and then you have to actually change the file type to fool the computer into recognizing it as an actual molecular model file. All right, let's go back to my list. And I'm going to show you the last of the small molecule databases that I wanted to show you. This is a database called Zinc, and it has over 3 million different compounds. Click here, and it brings up the web page for this thing called Zinc. I haven't actually looked at it. Let's see, it says it's provided by laboratories at the University of California, San Francisco, so it is something that is like an academic database. But I've explored in here a little bit, and it's really interesting. It almost seems like an online catalog for ordering quantities of chemicals from different vendors. And so I believe that scientists, who are involved in any kind of chemistry research, or possibly industries, that actually manufacture chemicals, can locate chemical compounds here in the Zinc database. And then it actually provides links to vendors where you can order this stuff. Now, I don't think that the average person can order stuff, especially if it's something like methylamine, which is a precursor for methamphetamine. It's not like you can just order a railroad car, tank car full of something like that. But researchers and people in academia and people in industry who have a legitimate reason to order this kind of stuff can do so. Anyway, that's just a bit of a tangent, but that's really why this actually exists. I just noticed this says there are 230 million searchable or purchasable compounds. And then down here it says 750 million purchasable compounds. So definitely got to update my website, because there's a lot more in here than I realized. To actually search for something, you click on the Substances menu, or Substances tab. And I'll come back to this thing on the left in a minute. But for the most part, you can just search for substances here in this text box. I'm going to look for bilirubin. And I'll click search. I noticed this earlier today. It was taking its time, but it should work eventually. Hopefully. It worked earlier today. Whoop, didn't like that. All right. I'll try that again, make sure I spelled it right. I don't know if it's just the server's overloaded or the internet's not happy at the moment, but we'll give that one more try. There we go. All right, so similar to one of the other databases I showed you, it returns a whole lot of different things that happen to have the word bilirubin in them, but many of these things are not what we want. They're all modified versions. Over here is just plain old regular bilirubin. So we can just click on this Zinc ID number, and this may take a minute. Oh, good, that actually went fairly quickly. And once again, this gives you a little bit of information about it. This SMILES string here, this long, crazy looking thing, it's similar to the IUPAC naming convention, but it's a code that actually describes the structure of the molecule. I've never really tried to learn these things. They look kind of odd. Down here, you actually get your list of vendors, places where you can buy bilirubin in quantities, which is kind of interesting. But there are a couple of places here where you can download a 3D model of the structure of bilirubin. Right here near the top, over on the right-hand side, there's something that says download. And once again, as with one of the other databases, the SDF format is the one we want. So let me choose SDF. And once again, it's thinking and taking its time. There we go. Save the file. I'll open that for you in just a minute, but let me show you that there's another place where you can also download 3D models. Should be exactly the same model, but it's just going to be in a different file format. If we scroll down a little bit, there's something here that says available 3D representations. And at the far right is another download link. Looks just like the other one. But when I click on this option, it gives us some other choices. And I'm going to choose the Mol2 format, which I know is going to open in a program like Chimera. It's actually trying to download a Mol2 with the GZ ending on the file. That means it's a gzipped file or a compressed file. So on my Mac, I have it set up that anytime it downloads a compressed file, it automatically decompresses it using this archive utility. So I'm going to go ahead and click OK. And it pops up the downloads folder. So here's the first one that I downloaded in SDF format. Let me double click that one. And it pops right up in Chimera. So this is the structure of bilirubin. I can tell right away that it's missing hydrogen, so I can go into structure editing and add H and see if this works. Yep, and it adds the missing hydrogen atoms. Let me close that session. And here's the gzip file, the Mol2 gzip, and then the one that's actually been uncompressed or extracted. If I double click that, same thing, opens right up in Chimera. Now this is interesting, this one actually shows the hydrogens automatically, so I don't have to add them. Let's go back to this page from Zinc. So I got here from the main Zinc page just by clicking substances. So what I did previously was I just did a text search up here in the upper right. This is a really interesting feature of Zinc, is that you can actually draw your own molecule and then it'll search for that molecule in the database. So if I choose one of these rings, like this happens to be a benzene ring, I can click with that. And then I can add some bonds on here. And then I have atoms on the left. You know, I have some standard ones like carbon, nitrogen, oxygen. I don't know what that one is. I can't even read it. Fluorine, chlorine, bromine, and so forth. I believe there's someplace I could click to get access to all of the other ones, but let me add some chlorines. I'll put a chlorine here, at that bond, and a chlorine here. And now I can search, but let's see. Don't need that. All right, I'm trying to remember where I go to actually search this thing. You can see one thing up here, it's actually added this thing called a SMILES string in here. Trying to remember how to do this. I don't know if, I don't think I, oh, there we go. It apparently when I clicked one of those other things, it did actually run the search. it's saying para here. This is actually a molecule. Surprised it's not giving the full name of it, but this is para-dichlorobenzene. You've got the benzene ring in the middle. You've got two chlorines on opposite sides, which are what we call the para configuration for things that are attached to a benzene molecule. And this is the stinky stuff that you find in mothballs when people put clothes away for storage and they put these mothballs in there to scare away the moths. That's what this stuff is. But anyway, that's just a simple example showing how you can draw a molecule. So if you have a picture of something and you have no idea what it is, you can actually come in here, draw it, search for it, and not only will you get a 3D model of it, but you'll find out more information about the molecule itself. All right, so those are my 3 favorite small molecule databases. Some of these other ones are not so great. At least one of these, I think it's this ChemExper chemical directory. I've had issues with that in the past where I download a molecule and it does give me a model, but it's a flat model. It's not actually a full 3D model, you know, with full 3 dimensions. So that could be a little bit of a problem, but you should be able to find just about anything you're looking for from those other databases, PubChem, ChemID+, or Zinc. So let's come down here to some of these macromolecule databases. And the first one I'm going to show you is by far the most popular. This is one that just about everybody in the medical illustration field is you know, somewhat familiar with. This is the Protein Data Bank. And the full name of it is the Research Collaboratory for Structural Bioinformatics, or RCSB. And I know that you can just click, I mean, you can just type in rcsb.org and it will automatically take you to the Protein Data Bank. There is a ton of information in here. Besides being able to search for individual proteins and download their 3-dimensional structure, there's also a ton of really valuable educational information in here. For example, because of the current virus pandemic, they've actually put up a link to a whole set of resources about the coronavirus structure. And one thing you have to be aware of is that none of these links are going to take you to a full 3D model of the virus. The links are going to take you to the individual protein components that make up the virus, such as this thing here, PDB structure 6LU7 is the high resolution crystal structure of a particular protease, a particular enzyme that's part of the virus structure. There are some illustrations and some 3D models and things, like here's David Goodsell's painting of the coronavirus. And down here, he actually identifies the names of some of the proteins that are found in different parts of the virus. Goodsell also wrote and illustrated a Molecule of the Month feature. I'll show you that in a minute, but this is about that protease, that enzyme that's found in coronavirus structures. And a bunch of other stuff in here. There's even a little picture you can download if you want to color your very own coronavirus. Let me go back to the main page. There is a link here, looks like a little chalkboard, PDB 101. This is where most of their educational resources are stored. If I click on that, there are a number of different things here, including a feature called Molecule of the Month. This is written and illustrated every month by David Goodsell. And if you look over on the right, you see the archive of past issues of the Molecule of the Month. And they're organized into different headings here, such as health and disease. So in here, you could look up, and there's probably several entries about HIV, several about diabetes, several about cancer. Let's explore the diabetes one real quick. So here are some different Molecule of the Month articles, all related to diabetes. And if I expand that, you'll see all of them. So there's one about designer insulins. Here's the structure of the glucagon receptor. Here's the structure of insulin. Insulin's actually a small protein. The insulin receptor molecule, the glucose transporter molecules that actually allow glucose to enter into the cell. So a whole bunch of excellent resources. If you were, let's look at the insulin receptor here. If you wanted information about, what's the structure of this molecule and how does it function, and if you read the text that goes along with the illustration, you'll see that Goodsell actually gives you links directly to the protein models in the protein data bank if you wanted to be able to download all or part of this molecule to create your own illustration. Also in PDB 101, down at the bottom are links to a couple of galleries of molecular art. There's a gallery of artwork by Irving Geis, who I've mentioned a couple times earlier in the course. He was the one who illustrated the three-dimensional structure of the first two proteins that were discovered. He illustrated John Kendrew's model of myoglobin, and he illustrated Max Peritz's model of the hemoglobin molecule. And he did a whole bunch of cover illustrations for Scientific American, all on different molecular structures. And then over on the right is a link to an archive of artwork by Goodsell. All right, but let's go back here. We're back here at the main page of the Protein Data Bank. And I'm going to show you a little bit about searching this, although I'm not going to go into quite as much detail as I normally do, because literally just a few weeks ago, the AMI hosted a webinar by Veronica Falconieri. I believe she used to work at the Centers for Disease Control or the NIH or one of those government labs. And it's pretty obvious from listening to her that she knows a lot more about the Protein Data Bank than I do. So I thought I would share her webinar with you. It's 1 hour long, so we'll do that after I finish talking. I'll show you her webinar. And her webinar is a beginner's guide to the Protein Data Bank, so she'll walk you through in detail about a lot of the different things you can do with this Protein Data Bank. But let me show you a little bit about what it does. Similar to a lot of the other databases, you have just a text field here where you can enter a bunch of stuff. You could put in an author's name. You could just put in a name of a particular molecule. Like I'm going to put in hemoglobin. And just click go. And it's going to return a list of everything in the Protein Data Bank that has something to do with hemoglobin. Now, it's very important to recognize that a lot of what's here is not actually molecules of hemoglobin. I mean, most of these are, but there was one up here, the structure of the T. brucei haptoglobin hemoglobin receptor binding to human haptoglobin hemoglobin. Now, there's a mouthful. But I can tell just from reading that, even if I don't know what half these words mean, that this is not just a plain old model of hemoglobin. Now this one is deoxyhuman hemoglobin. It's from a human, it's hemoglobin. Deoxy just means that it doesn't happen to have the oxygen bound to it at the moment. So this is a pretty plain normal structure of hemoglobin. So if you wanted to download this one, this would be a good choice. Things like recombinant hemoglobin means that it was probably manufactured using recombinant genetic engineering. There's some more recombinant ones. Cross-linked, not sure what that's supposed to mean. So a lot of these, you just have to kind of read the titles and see if you can figure out which one is just plain old hemoglobin. Now here's one that's deoxyhemoglobin. That's pretty basic, but it's from a hagfish. It's probably not that different from the human one, but it might have some subtle differences. So if you're really looking for an illustration of human hemoglobin, you want to download something like this. Now you can actually dig deeper into these results. Notice up here, there's actually 752 different entries just for hemoglobin or things related to hemoglobin. So I might want to narrow that down just to make my search easier. So over here on the left are all sorts of ways that you can refine the search. So I could search just for 297 different versions of human hemoglobin, or I could search by other species. I'm actually going to search for Equus cabeus because if you know a little Latin, you know that means horse. I'm actually going to try to find Max Perutz's original model of horse hemoglobin. So I'm going to click on horse here. That also helps there's only 20 results, so it's going to narrow it down a little bit. So now I've only got 20 results. I can narrow this down further by experimental method. I know that Perutz used X-ray crystallography. You can narrow it down by resolution. Remember, when you're talking about resolution of an X-ray diffraction image, less is more. A small number is a higher quality image. I can also narrow it down by when the model was constructed or when it was deposited into the protein data bank. And since I know Perutz was the very first one to solve the structure of hemoglobin, it's more than likely going to be one of these that was deposited before 2000. And sure enough, there are two here. The structure of horse methemoglobin at 2 angstroms resolution, and then 3-dimensional Fourier synthesis of horse deoxyhemoglobin at 2.8 angstroms resolution. Well, the 2 angstrom one is going to be better resolution, but I'm not 100% sure what the methemoglobin is referring to. This is just deoxyhemoglobin, so I know that means that it's plain old hemoglobin, just doesn't have the oxygen bound to it. Even though the resolution is not great, I know that it's a pretty good model. And Perutz actually did his initial research on hemoglobin in the late 50s and early 60s, but this one wasn't deposited until 1970. So let me go ahead and click on this one. I'm just doing this to demonstrate that there's all sorts of different ways to narrow down your search results. So this 2D-HB is the PDB ID number. So that's a unique number assigned to this particular model. There's some information about it here. It's an oxygen transport molecule. It's from the species Equus cabeus. This is a title of an actual article that described the molecule. And then down here, we're going to look at this section called macromolecules. And this is something that Veronica is going to explain better in her webinar later. But these little bars that you see down here are representations of how complete the molecule is and also how complete the genetic code is. Have we actually been able to determine the sequence of the gene that codes for this protein? So down at the bottom, this blue bar is a representation of the protein. And the fact that almost 100% of it is solid blue without a whole bunch of gaps in it, that tells me that this is a very complete model of the hemoglobin protein. You will sometimes come across models that have great big gaps in this blue bar, which tells you that it's not complete. A lot of these other bars, I'm not as sure what they mean. There is one here that's called secondary structure, or sec structure. The red bars indicate where there are alpha helices in the protein, so you can actually kind of look at it and just see where the different secondary structures are. Something else I should point out here is that everything you're seeing here, all of these bars are for what's called entity ID number 1. And if we look down below that, it says hemoglobin alpha chain, or chain A. It turns out that a fully functioning hemoglobin molecule has 4 chains. There are 2 alphas and 2 betas to make a functioning hemoglobin molecule. So everything here that you're seeing described is just for the alpha chain. And then if we scroll down a little bit, we see entity ID number 2, hemoglobin beta chain, or B. Now this happens to be turned off at the moment, but there's a little button here I can click that expands what's called the protein feature view for the beta chain. And once again, if you look at the blue line across the bottom, you can see that it's a very, very complete model of the protein. One more thing I can look at down here is an entry called small molecule. If there are any kind of, just pausing for a second because my computer told me my internet connection was unstable. That's not good. Hopefully it won't crash. This thing called small molecules shows you any other molecules that are not actually part of the protein itself that may be included in the model. Now, in the case of hemoglobin, you have the protein component itself, which is known as the globin portion of the hemoglobin molecule. But then you also have something called the heme group. The heme group is a ring of carbons. You can see a little representation of it over here on the right. And the iron atom of hemoglobin is actually suspended in the middle of that ring. And it's the iron atom that actually bonds with the oxygen to transport oxygen in the bloodstream. So these heme groups are the portion of the molecule that actually, holds the oxygen, but a heme group is not a protein. It's not actually part of the protein itself. It's something that's embedded within the alpha and beta chains of the hemoglobin molecule. So it's listed here as a ligand, meaning, something that's bound to the protein. If you look at other protein data bank entries, sometimes you'll find very large numbers of small molecules that happen to be included in the molecular model file. If you downloaded something like an acetylcholine receptor, if you're lucky, you'll actually get a model where the acetylcholine is actually included right in the model, so that you could see where the acetylcholine neurotransmitter actually binds to its receptor. So sometimes, these small molecules are really important. Certainly, the heme group is critically important for hemoglobin, but just looking and reading about the small molecules may be important because it may be that there's the actual ligand that binds to a receptor may actually be included as part of the model.
Hey, Jim.
Yeah.
What did you say the gaps in the blue bar represented when you were talking about the entity entries?
Yeah, the entity entries and this protein feature view. the gaps represent small segments of the protein that they may not have been able to determine experimentally. Now, oftentimes, they know the linear sequence of amino acids, but sometimes some of those individual atoms don't show up in their X-ray study, and so they're a little uncertain about exactly where the atoms belong, but they still know the sequence. So they know there's got to be, you know, a proline or a histidine or you know, a leucine or whatever in that position, just because they know, you know, the sequence of amino acids. But it may just be that it didn't really show up in their 3D model. So they probably filled in the gaps with their best guess as to what it looked like. There is something else later in the course. I'm not sure if I'll have time to address it, but there's something called a homology model, which is a lot of guessing. It's a model of a protein that's not really solved by X-ray crystallography, but it's our best guess because we know the structure of things that are similar to it, so we can kind of approximate what the structure would be. So, I mean, this model is extremely complete because there's only some tiny little gaps. And my guess is that they did a pretty good job of filling in those gaps. But you will find other models in the protein data bank where there's big chunks of this blue bar that are just missing. All right, so let's come back up here to the top of this entry for hemoglobin. And we'll look over here on the left. There's a picture of the model, but there are actually a couple of different pictures that you can look at. There's little arrows here that let you scroll back and forth between these things. There's actually only two different versions. This is a picture of what's known as the asymmetric unit. Now, when they did this crystallographic study, apparently they really only solved the structure of one alpha subunit and one beta subunit of the hemoglobin molecule. So the asymmetric unit is really only half of the hemoglobin molecule that was, you know, studied in this particular X-ray crystallographic study. If I click the arrows and change to this other view, this other picture, this is what's known as the biological assembly. So what they've done here is they've taken the one alpha chain and the one beta chain, and they've simply copied them and flipped them more or less mirror image or rotated them 180 degrees to create the other side of the molecule. So this biological assembly actually shows the fully functional hemoglobin molecule with two alpha chains and two beta chains. So whenever you're looking for something in the protein data bank, if you have a choice of downloading something called the asymmetric unit or downloading the biological assembly, almost all the time, unless you've got some good reason to not do it otherwise, always download the biological assembly, because that's going to be the complete model with the full structure of the molecule. Just below these two-dimensional images, there's something called 3D view, and we can click to view the three-dimensional structure of the molecule right here in the web page. This brings up a molecular viewer, which is embedded, you know, right in the protein data bank web page. And I can click on this and rotate it around in 3 dimensions. And over on the right, there's a bunch of little pop-up menus that allow me to change what we're looking at. So right now, by default, it's showing us the biological assembly. But I could switch that to asymmetric unit, and you see what we end up with is only half the molecule, one alpha and one beta chain. So let me go back to biological assembly. I can also change the style. Now, they have some different names for things. Actually, Backbone, I believe, is, it looks like a typical Backbone model. You can see the heme groups floating in the midst of those backbone models, by the way. I could switch this to Cartoon, which is their name for a ribbon model. I could even add a surface. Let me go back to the cartoon or ribbon view. I can also change coloring. For example, I could change it to coloring by chain. So now you can see that there's 2 red chains, and that would be part of the beta chain. There's a B in that, that little text field that pops up. And if I hold down, or if I hover over the blue ones, there's an A. This is part of a valine residue. It's valine at the number 70 position in the molecule. The A there indicates that it's in the A chain. I can also change the representation of any ligands that are present. Right now, by default, the ligands are being shown as ball and sticks, but I could also change that to space filling representation of the heme groups, or I could just turn them off completely. The last thing I wanted to show you here in the protein data bank is in the upper right corner of this 3D model window, or even if we go back to just the text page, there's always a link here in the upper right to download files. And this is how you're going to actually retrieve the 3-dimensional models. So if I download files, I can download it directly in PDB format or in a gzipped compressed PDB format. But that is going to give you by default just the asymmetric unit, just in this case, just half the molecule. It may be better, and it's almost always a better idea to scroll down to biological assembly to make sure we're getting the full thing. So I'm going to download the biological assembly. Apparently that's a compressed file also. It's a gzipped file. So I need my decompression software to decompress that. And it pops up here in my downloads folder. It's 2D HB PDB1. Double click that, and there it is in Chimera. And here in Chimera, we could select this by chain. If I say select chain A, that's going to be the alpha chain, but there's 2 alpha chains. So I could select one alpha chain and give it a color. I could go back and select the other alpha chain, give it the same color. Select one of the beta chains, give that a different color, and select the other beta chain and give that color. So now you see the complete structure of hemoglobin with 2 alpha and 2 beta chains.
Hey, Jim, quick question.
Yeah.
Is there ever like an undo that you're able to do on Chimera, or is it kind of just like go with what you have?
Yeah, I've never found an undo.
Okay.
In
I was
here.
just curious.
Yeah, like, so if you change something to a different representation and you didn't want it, you just kind of stuck with having to go back and change to a different representation.
Okay, gotcha. Thanks.
Yep. So I'm going to deselect. Remember that there are these heme groups in here. Now, right now, they're just colored the same as the chains, but this is a little trick that I showed you on Monday. If I want to just select the heme groups and maybe change the representation, I can go to structure or select structure protein, which is going to select all of the protein components. But then if I say select and invert all models, that selects everything that's not protein, which in this case just happens to be the heme groups. So now that only the heme groups are selected, I could change them maybe to a sphere representation, and I could color them by element to get like a CPK representation. All right, so let me close that. Okay. So I'm going to go back to my web page, and I'm going to scroll down here to one of my personal favorites, is the OPM database, the orientation of proteins in membranes. I've already mentioned this to you, and I even showed you an example of a molecule that I downloaded from here. You may not remember it, but this is a database that lets you download proteins, but it shows you the two layers of the lipid bilayer of the cell membrane so that you know exactly how this molecule fits into the cell membrane. Now, of course, this only works for molecules that are actually embedded, proteins that are actually embedded in a membrane, but that includes an awful lot of receptors and channel proteins and things like that. So it's a very handy database to use. So here we are in the OPM database. There are ways of digging down into the database, but I'm not going to show you any of that. We can just use the simple search feature here in the upper right. And I'm going to look for a particular acetylcholine receptor. I should be able to just enter the word acetylcholine, and it's going to find every protein in here that has something to do with acetylcholine. And sure enough, we get a list of 36 different proteins that have something to do with acetylcholine. There's a bunch of information here, of course, organized into different columns. The family column tells me what category of protein does it fall into. And there are many different categories of proteins, and if you haven't taken a little bit more, cell biology or molecular biology or something, you may not be that familiar with some of these. But it's a good idea to try to familiarize yourself with some of these things. There's a whole group of very important signaling proteins in all living things that are called G-protein coupled receptors. What that means is that you've got a receptor molecule, a receptor protein that binds to some ligand, but then that activates another protein on the inside of the cell membrane known as a G protein. And it's the G protein that then causes some action to occur, some reaction to occur within the cell. So the receptor on the surface is coupled with a G protein inside. So that's where this name comes from, G protein coupled receptors. So there are some acetylcholine receptors that function this way, where the acetylcholine binds to a receptor on the surface that triggers a G protein inside the cell, which then leads to a whole cascade of things inside the cell. But if I scroll down, I find that there's another whole family of proteins. There's a category of proteins called ligand-gated ion channels. In this case, if you read the full name, it would be ligand-gated ion channels of neurotransmitters. There are other acetylcholine receptors that when acetylcholine binds to them, the protein itself is actually a channel. Typically, it's a sodium channel. So once acetylcholine binds to it, there's a small change in the shape of the protein. It opens up a pore in the middle and allows sodium ions to rush into the cell. This is the kind of acetylcholine receptor that you typically find in the synapse, where you have acetylcholine released by the presynaptic cell. The acetylcholine flows across the synaptic cleft and binds to these ligand-gated ion channels on the postsynaptic cell. That causes the sodium channels to open up on the postsynaptic cell. All that sodium flushes in, and you get the generation of an action potential, causing that second neuron to fire as a result. So I'm going to download one of these. I'm going to click directly on its name. Let's see, is that the one I want? Let's see, I'm going to pick this one just because I've looked at this one before. There's even within these ligand-gated sodium channels, you can see there's a bunch of different ones. So when I click on that one, we get just a little bit of information. It's transmembrane protein, meaning it's embedded in the membrane. My internet connection is slowing down again. Hopefully this still works. It's a bunch of alpha helices that cross the membrane. And then it's in the family of ligand-gated ion channels of neurotransmitter receptors. This one happens to be human. There's a little picture of it here. And we can download the file by clicking over here, download OPM file, 6pv7.pdb. The 6pv7 in this case is the PDB ID number. So this is a molecule that you could find in the protein data bank. However, the benefit of the OPM database is that it includes these two layers of balls that allow you to see both the extracellular and the cytoplasmic sides of the plasma membrane, or the lipid bilayer membrane of the cell. So that's the advantage of the OPM database, is that even though it uses that same PDB ID number, downloading them from here gives you that additional information about where the protein is actually embedded in the membrane. So I'll click here to download the file. It's going to launch Chimera automatically. That's okay, because I was going to do that anyway. And here is the molecule. Now, when I rotate around this molecule, I can see that the protein itself is represented as a ribbon model. But there's a whole bunch of other junk in here. There's a whole bunch of little stick models of something else. Those are all ligands. Now, I don't think those are acetylcholine. If I went to the actual protein data bank entry for this model, for that PDB ID number, whatever, I can't remember what the number was off the top of my head, but if I looked up that PDB ID in the protein data bank, it would tell me what all of these ligands are that are attached here. I want to get rid of those, because if I'm creating a picture of this molecule, I don't want to accidentally include images of those ligands. So under my select menu, I can actually go to select structure and ligand. So any ligands that are present can easily be selected from the select menu. You notice that they all get highlighted in green. I can go to actions, atoms and bonds. I could hide them, but better yet, I'm just going to delete them completely, because I know I'm not going to use them for anything. So they're gone. Now what I'm going to do is I'm going to select structure protein, So I've selected the whole protein. Now I'm going to select invert all models, which means if there's anything else left in this model, it's going to select anything other than protein. I did get rid of the ligands, so it's not going to select them. They're gone. But if there's anything else in here besides protein, this will select them. So I just said invert, and it doesn't look like there's anything there. But I know just from experience using these OPM files that there really is something here. I'm going to say action, atoms and bonds, and show. And sure enough, there really is something that was selected. They were just temporarily hidden. These are the two layers of balls that allow us to see how this protein is embedded in the cell membrane. I can change the representations to spheres so that I can see that more clearly. Now, the convention that the OPM database uses for these colors is that the outer layer, meaning the layer of the cell membrane that is facing towards the outside of the cell, is colored red, because the first letter of the word outer is O, and red is the color of oxygen in the CPK scheme, color scheme. The inside layer of the lipid bilayer is colored blue. That's because the word inside kind of sounds like it starts with the letter N, which it doesn't really, but you've got inside, so there's that prominent N at the beginning of the word, or near the beginning. And N is for nitrogen, and nitrogen is blue in the CPK color scheme. So for outside, blue is for inside. So this shows you exactly how the molecule sits in the lipid bilayer of the cell membrane. I've still got those layers of balls selected. I could invert again, which goes back to my protein, and I could add a surface to that protein. So now you really see what that protein would look like as it's embedded in the cell membrane. If I tilt it this way, you can look right down the center, and you can see the pore, the opening, where those sodium atoms or sodium ions would pass through to flow from the outside of the cell to the inside. If I tilt it up the other way, you'll see that the pore is visible down here on the bottom as well. I could also select the individual chains that make up the protein. I don't think that got one of them. Let me see here. There we go. The acetylcholine receptor is actually made up of quite a few different chains. So I'm just showing you that you can colorize all those chains independently if you wanted to. And I won't bother doing the rest of them. All right, so let me close that. All right, tell you what, why don't we take a short break, and then I'll come back and I'll just show you two more databases that should go pretty quickly, and then I'll play that webinar, the Beginner's Guide to the Protein Data Bank. So let's just take about 10 minutes. My computer shows 3:13, so we'll start about we'll call it 3:25. And I'm going to stop sharing my screen temporarily and pause the recording. and share my screen. And every time I do that, my chat goes away. It's not that it did much good, because Austin did send me a question by chat. That was his question about what do the little gaps in the blue bar represent, and I missed it. I missed his comment, but I'll try to keep my eye on that chat over in the corner of my screen. All right, so couple of last macromolecule databases that I wanted to share with you. There's one that's kind of relevant right now, the Viper database from Scripps. This is the Virus Particle Explorer. And this lets you download what's called the nucleocapsid of different viruses. Now, something I should warn you about. If you think you're going to go here and download an entire model of the coronavirus, the COVID-19 virus, or really any other virus, unfortunately, this does not have the full structure of any viruses. Instead, all it has is what's known as the nucleocapsid, which is a structure made of protein in the center of the virus that actually holds the nuclear material or the genetic material of the virus. There are some viruses that have a strand of DNA, a strand of RNA, but in either case, it's the nucleocapsid, this core of proteins that surrounds that genetic material. Most viruses then have additional stuff around the nucleocapsid. In fact, there's a lot of viruses that have a full portion of cell membrane, a piece of that lipid bilayer cell membrane surrounding the entire virus. And then there are other proteins packed inside that cell membrane, as well as various kinds of receptors and spike proteins and things on the outside. So if you look at any of the pictures of the coronavirus, what you're seeing is actually the exterior of the virus, which is really just a piece of that lipid bilayer cell membrane. And then you're seeing all these spike proteins and other things that protrude from the top. Most of those pictures of the coronavirus don't even show you the interior, so you're not even seeing the nucleocapsid. So the structures you can download from this website are really just that internal nucleocapsid that surrounds the virus's genetic material. Here at the top of the page, there's a link to find a virus. And brings up this simple form where you can just enter a name of a virus. And just for kicks, let's check. I've never actually looked for this one before. Let's see if there is a coronavirus. Usually, when you enter the name of a virus, you actually get a list that pops up immediately, and it says to scroll down to find the one you want. So I'm thinking that means it does not have a coronavirus structure in here, or COVID. No. There's circoviruses and things like that. All right, well, let me put in one that I know actually works. If I type in the word hepatitis, I get a number of different hepatitis structures. So let's download the hepatitis B viral capsid. And over here on the right, you can see a little thumbnail of what that viral capsid looks like. The small ribbon models that you see just above the view of the capsid, that's the actual protein that makes up the capsid. So there's a whole bunch of copies of this protein that are arranged in roughly a sphere. Many viruses, the nucleocapsid is a geometric structure known as an icosahedron, which looks kind of like a soccer ball. So it's just a little cluster of proteins that are repeated. I think it's 72 times. I may have the number wrong, but it's an identical cluster of proteins repeated a whole bunch of times around, basically in a spherical or sort of soccer ball shape. So what you're seeing on the right is just the individual protein, and then you're seeing what it looks like when it's all put together into the complete nucleocapsid. But remember what I said, that you're just getting this internal core structure that contains the genetic material. You're not getting the whole virus. You can actually download either the full capsid, the entire sphere, or a half capsid, which is basically just a hemisphere. I'm going to try that one. It's 2 megabytes, so it's not too bad. It shouldn't take long to download. I do know, though, once I try to open this, it may take a little while to fully open. So here it is. There's a 1QGT half vdb.gz, the gzipped version, and then I've got the expanded one. The VDB file type, it's really a type of protein databank file. It's a PDB file, but it gives it this special file format because it's coming from the Viper database or the virus database. If I double click on this, it'll launch Chimera and hopefully not crash my computer, because these virus particles are very large. Here it is. So you can see that what we've got is basically half of the virus nucleocapsid, and the little white and purple things are the individual proteins that are then repeated in this sort of soccer ball shape. So it's a very specialized database, but it's actually kind of cool to be able to play with these virus particles.
Is there a way to open 2 files in the same scene and orient them?
There is a way to open 2 files in the same scene. If I didn't keep closing session here, every time I was done with a molecule, I could just keep opening a whole bunch of them and have them just all jumbled on top of one another. I'm not sure I can tell you how to actually move them individually. That would be the problem, is you can open a whole bunch of them all at the same time, but whether you can actually open them and move one without the other, frankly, I haven't tried it. So there's a little task for you, Riley, is open.
I haven't figured it out yet either.
Yeah, I don't know if it's got something to do with, I mean, you'd think maybe it has something to do with selecting them, but I'm not sure that works.
Yeah, I think there's like a pivot, like you can set a pivot, but I don't know if that.
Yeah, like I said, I haven't really tried it myself. Would be interesting to try that, especially because there are some proteins where when you go to the protein data bank, you can only download a fragment of the protein. Sometimes it's very difficult to download the whole thing. I think Veronica's webinar will address this a little bit too, that sometimes when you go to the protein data bank and you download something, if you actually read the description of it, it'll say this is only the extracellular domain, the part that's outside the cell. Then there may be a completely separate PDB file for the transmembrane domain, in other words, the part that's embedded in the membrane. And then there may be even a third model for the intracellular domain. And without a whole lot of molecular biology, it may be hard to put those together. But if you could open all 3 of them in Chimera, maybe there would be a way to actually kind of stick them together to create one model of the entire molecule. But like I say, that's not something that I've tried myself. All right, let me go back to my list and show you one last database. This one is kind of fun, but it's also, quite functional. It has some real handy uses. Hopefully this still works. I didn't get a chance to check this one out before. This one's moved a couple times over the years. All right, so far so good. This used to be hosted from a university in Italy. Now it is, I forget where it is. If anybody knows where that is, that Catholic university, somewhere in Eastern Europe. This is I won't even call it a database because it's not really a collection of models. It's an online tool that allows you to enter your own string of nucleotide bases, A's, T's, G's, and C's, and then it will actually build a piece of DNA for you with that sequence of bases, and you can then download it as a PDB file and open it in your favorite molecular program. So from this main page, you click on the input form. And it asks for the name of the molecule, which is optional. You don't even have to put that in. Let me see what happens if I put in something like my DNA. And then you put in your sequence. So just put in A's and T's and C's and G's. I think you can put in up to 100, or no, up to 700, excuse me. And then, very important, you have to specify parameter, meaning specify exactly what kind of molecule do you want. And if you're trying to create an actual double helix model of DNA, you want to choose the last one, straight bDNA. There are a couple of different forms, naturally occurring forms of DNA. There's something called aDNA, which is very compressed and very tightly wound up. That's the one that people like Linus Pauling and Watson and Crick were working with originally, because they hadn't discovered the trick of hydrating the molecule when you actually did your X-ray crystallography. And so then Rosalind Franklin discovered that you had to perform the experiment under conditions of high humidity, because the DNA is normally immersed in water anyway inside the cytoplasm of the cell. So this bDNA is the form that actually exists inside the cell in that aqueous environment. So that's the one you want. So we've got our string of A's and C's and T's and G's. We've said we want a model of straight bDNA, and I click the little button that says send it. And immediately it says, here is your PDB file. I'm going to click on that, save it to my downloads. I'm not sure. It ignored the name that I put in, so there's probably no point in putting a name in. It didn't call it my DNA. It's just called mimeget.pdb. But if I double click that, sure enough, we get a chunk of DNA. And one of the two strands actually includes that exact sequence. Now, I don't remember what sequence I put in off the top of my head anymore, and it really doesn't matter, because it's rare that you would ever draw a DNA molecule and say, oh, this one has to have the exact sequence, A, C, T, G, T, A, because most people would look at this and they wouldn't know the difference. They just see a bunch of bases, in this ladder form. You remember last time I told you that you can go under Tools, Depiction, and Nucleotides, and bring up a bunch of different ways of displaying any of these nucleic acid models.
I figured out how to move it.
Did you?
Yeah, there's a mouse movement tool, and then you can select move selection, and you can select individual molecules and position them individually and move them in space, like translation, rotation.
Oh, cool. Is that something that's actually under the tools? Oh, is it the movement?
Yeah, somewhere in there.
Movement mouse
Yeah,
mode,
yeah,
one of these
that's
things. Yeah, okay.
Movement mouse mode.
I'll have to play with that and give it a try.
Very helpful.
Okay. Good to know. Thank you. All right, so I think I'm done demonstrating different databases. So I'm going to quit Chimera now, close my downloads, get out of this window. And here we are at what's called the AMI Members Hub. I have to tell you a little secret, and hopefully nobody's going to turn me in. The webinar that I'm about to show you is really intended for AMI members. AMI members could sign up to watch the webinar live, and I watched it live a few weeks ago. And then the webinar is then saved in something called the AMI Members Hub, which is a members only section of the AMI website. So the content in here is supposed to really only be accessible to AMI members, but I didn't really have time to check with them and get their permission, but I don't think they would have an issue with me, sharing it with the students, especially because, once you see some of the stuff that they have available, it might actually encourage you to join as a student member now, and then eventually in the future join as a professional member. So there's actually a whole bunch of stuff in here. I'll just take a little tangent here. There's something called the Hub Forum, which is the AMI's listserv. So I get emails, every day. Lately, there's been a ton of them about all of this virus stuff. There's some, AMI news, association resources, which is just a lot of stuff about, well, there's a whole link in here about board certification, which is a useful thing. But there's also stuff about the committees and the governance of the AMI. Business resources is very, very useful because they have job listings. They also have a whole section of sample contracts and forms. This is very useful. I've used this a number of times to download sample contracts for situations that I don't normally encounter. I mean, I know enough about the copyright law and so forth that there are some contracts that I could write up myself, but there have been a few times where I've had kind of a unique situation where I really needed some advice, and I was able to come in here and find a sample contract that actually suited my needs. I just changed the language very slightly. And then here's online education. They have announcements for new webinars, and then they have the whole archive. And this is the most recent one that I'm going to show you, the Beginner's Guide to the Protein Data Bank. But they have some here about using Affinity Designer, using something called Redshift in Cinema 4D, educational research for medical illustrators, organic histology, so drawing really natural looking cells and tissues. The AMI does a salary survey every 4 years. And this was just a webinar talking about some of the data that they learned from the most recent salary survey. And, some Photoshop tricks and techniques and so forth. So let me share this one with you.
Hi, everyone. Welcome to the webinar. My name is Seemay Johnston, and I'll be introducing our speaker, Veronica Falconeri, in just a moment. First, I'd like to note that this webinar is organized by the Online Learning Committee. We provide opportunities for AMI members to enrich their knowledge and earn CEUs. We're always looking for volunteers, so if you're interested, please email hq@ami.org. So now I'm pleased to introduce Veronica Falconeri. Sorry about that. My computer's making noises. Veronica will be presenting a beginner's guide to the Protein Data Bank. Veronica is the principal of Falconeri Visuals, a medical illustration and animation company specializing in molecular visualization and research. Prior to launching Falconeri Visuals, Veronica worked as an illustrator, animator, and project manager at the National Cancer Institute for a lab that studied macromolecular structure using cryo-electronic microscopy, cryo-EM. She is a published author in journals including Science and Cell. She received her MA in Medical and Biological Illustration from Johns Hopkins University School of Medicine. Also, a quick note, Veronica's prepared a handout that you can download in the handouts panel of the GoToWebinar interface. Also, she'll be presenting for 45 minutes, roughly 45 minutes with a 15-minute Q&A session. So feel free to type in your questions in the questions panel. So with that, I'll hand it over to you, Veronica.
Awesome. Thank you for the introduction.
Let me find you on the panel. There we go.
Okay, so again, thank you for the introduction. And as Sime said, I'm Veronica Falconeri Hayes, and I'll be walking through today a beginner's guide to the protein data bank. And the reason why I put together this webinar is just because I remember when I first got started with molecular visualization, navigating the protein data bank was like pretty hard to do. But once I spent a couple of years actually working in a lab with scientists who work on molecular structures and kind of, you know, getting their perspective, that really helped me understand the protein data bank. And then just after years of practice of accessing it and doing research in it, you know, I can use it a lot more smoothly. So I'm hoping to share some wisdom from that experience today. So first, most basic thing, what is the protein data bank? And it is primarily a repository for experimental results. So when scientists determine a 3D structure, they need to deposit those results somewhere, and they do that within the protein data bank. And that means that the primary purpose of the PDB is for scientists to communicate their findings in detail to other scientists. And this is really important to remember. So when you end up looking at a PDB entry and you look at all the information there, you have to remember that this is coming from one structural biologist, and they're trying to communicate to other structural biologists. So that's why all of the information is organized the way that it is. So it's really important to remember our protein structure terminology when we're looking at these entries, because that's how it's all, how all of the PDB is organized. So first, let's start with the term biological macromolecules. So macromolecules can be any molecules composed of a very large number of atoms. There's no particular number cut off here. So we just see the term macromolecules a lot within the PDB. Typically, these biological macromolecules fall into 4 categories. We have proteins and nucleic acids, which we see in this image here, where we've got DNA, some RNA, some protein. There are also carbohydrates. So here what we're seeing is a viral spike on the surface of HIV, and the red molecules are carbohydrates that are on top of that spike. So the spike is protein, and then there are carbohydrates attached to the surface. And then, of course, there are lipids, which we typically think of in lipid bilayers, but these can also sometimes be found, you know, connected to proteins. Now, within the protein data bank, there are primarily proteins and nucleic acids, and all of the terminology for how the entries are structured is based on proteins. So let's review some of that terminology. So we're primed and ready to jump in. So the basic unit of protein structure are amino acids. I also know they are also referred to as residues sometimes, and I'm saying that because a lot of times I will use those 2 terms interchangeably. So if you hear me say residues, I just mean amino acids. So amino acid structure is fairly simple. We start with, we have a nitrogen on one end, the amino group, and that's why it's blue. A carbon backbone, and on the other end, there's an oxygen attached to a carbon or a carboxyl group. And off the side of this is a side chain, and that varies. So that could be one of 20 different little structures. So that's the basic structure of an amino acid. And if you line these guys up and you attach them with a peptide bond, what you end up creating is a polypeptide chain. And that polypeptide chain has an N terminus on one end, and that's where the amino group is sticking out, and a C terminus on the other end, where that carboxyl group is sticking out. So the N terminus to C terminus is an important terminology to remember, because in the PDB, we see all the sequences listed in order from N terminus to C terminus. So it's important to remember which end is which. So that brings us to the primary structure of proteins. The primary structure is just the order of the different amino acids from the N terminus to the C terminus. So that's what we're looking at here, is the primary structure of a protein, where each of the amino acids has been indicated by a single letter code. Amino acids can be encoded either by a 1-letter or a 3-letter code, depending where it is. In this case, we're just looking at a single letter code. So each of these guys is amino acid starting N terminus here at 0, and C terminus over here around 142. Now, secondary protein structures, if you take that polypeptide chain, that string of amino acids, and you fold it in 3D space, there are certain patterns that happen to occur. And if you're looking at a protein here in ball stick model, they're not particularly evident, but that's why scientists have developed the ribbon or cartoon structure to make it more obvious. So what we're looking at here are some common secondary structures. So in magenta, there is the alpha helices, and in yellow, we have beta strands. Now, there are also things like beta sheets, where you have a lot of beta strands lined up next to each other. And then there are also loops, are these like white areas that don't necessarily fall into becoming a structure that's either a helice or a sheet. Tertiary structure, not as important, doesn't pop up as much within protein structure terminology that you need to know, but it's basically how these secondary structures are further folded. So if it's maybe this tertiary structure would be, these two alpha helices are next to each other. That's a description of the tertiary structure. And then there's quaternary structure. So that's the organization of multiple polypeptide chains. So here we're looking at an antibody, and it is actually made up of four different chains. Or actually, I should say 2 different chains, 2 copies of each. So we have 2 light chains in the yellow and the purple, and then 2 heavy chains, the red and the blue. So this would be a tetramer. So it's important to keep in mind or be familiar with, dimer, tetramer, trimer, heterodimer when looking at the PDB, because sometimes you'll see structures described, this protein is a heterodimer. So you need to be able to kind of look at that and be like, okay, so I should expect to see 2 different proteins stuck together, heterodimer. And then on top of protein structure, or like kind of the final level of it, are post-translational modifications. So these are things that are added to proteins or changes that are made after they're assembled by the ribosome. So for example, here, if we look at this image again, these glycosylations, which are the sugars on the surface of this spike protein, those are an example of a post-translational modification where a carbohydrate has been added on to the protein. And if we go back to our antibody and look at it with a little bit more detail, we can see disulfide bonds. That's another example of post-translational modification where these 2 sulfur-containing amino acids have been connected between two different polypeptide chains. So there's a lot of different post-translational modifications that can occur. So just kind of keep an eye out for them, because sometimes they're really important to protein structures. All right, so that brings us to the Protein Data Bank, which is home to, as of sometime in February, over 160,000 structures. So where are all of these structures coming from? So most of the Protein Data Bank comes from the field of structural biology. So when we're looking at cells and tissues and things like that, standard light and electron microscopy methods can be used. So looking kind of at this level. But when you want to look in detail at the 3D structure of molecules and how they're interacting, it takes more specialized methods. And that's the field of structural biology, is how do we look at these structures in such high detail? And there are a couple methods that make up the bulk of the Protein Data Bank. So most of the structures within the Protein Data Bank come from X-ray crystallography. Then we also see a fair number that come from NMR, or nuclear magnetic resonance. And then there's a growing segment that's coming from electron microscopy techniques. So what we'll do is just do a brief overview of each of these methods, so you can get a feeling for where the data that you see in the PDB are coming from. And that kind of also impacts the way that you read those PDB entries. So first, X-ray crystallography. Scientists get a solution of their protein, manipulate it into crystallizing. They get a crystal and they can look, these are all protein crystals. Shoot an X-ray through it, it refracts in a certain way and creates a diffraction pattern. And from this diffraction pattern, they can calculate back and tell where the electrons are most dense in 3D space. And they get a density map, which is volumetric data. And then they take the primary sequence, or the primary structure of the protein, and they are able to use that to fit a model into this 3D density map. And that is what is actually known as your PDB. So it's a model of all the atoms in the protein fit in 3D space according to the map. And we'll get back to that again later. Now with cryo-EM, they take a different approach. So instead of crystallizing a protein, they take a purified protein solution, they spread it really, really thin on an object known as a grid until there's only one molecule thick. So that's what we're looking at here, is if we had many copies of this green molecule and it's in this really, really thin layer, and all of these proteins are at slightly different angles. What scientists then do is they plunge freeze this and they put it under the microscope. They take an image and then through computer processing, they pick out thousands of images of those proteins, all at slightly different angles, and average them together to get to their protein structure. So they also get to a density map, which is what we're looking at here, but just through different means than they would with X-ray crystallography. Nuclear magnetic resonance is inherently different. NMR actually measures the distances between atoms, and the output from that is an NMR spectra. So the output for a really small, simple molecule like ethanol looks like this. And you can also use the same principle when looking at proteins, but because the spectra get fairly complicated, NMR is limited to small proteins, so ones that are about less than 200 residues. However, there is a benefit of this, and it's that we can actually get multiple conformations of a single protein. So what we're looking at here is an NMR structure for insulin, where with the PDB entry, they've also deposited, I think, about 20 conformations as standard. So you can see, one conformation I've highlighted here, but then there are many, many others that are all also normal conformations for insulin. So we can see some of the flexibility of the molecules. All right. So because all this PDB data comes from experimental sources, there are experimental artifacts very frequently in PDB files that we have to keep an eye out for. One of the most common, antibody fragments. So this will show up in both, cryo-EM and X-ray structures where they either needed to use an antibody binding to their protein to make it more stable or to make it larger. So in order to crystallize it or in order to see it more clearly under the electron microscope. There are also engineered mutations. So sometimes they're stabilizing the protein by changing an amino acid somewhere, or they're making it inactive so it isn't undergoing its enzymatic activity in some way that would prevent them from doing their experiment. Sometimes there are chimeric proteins, which is what we see here, where they're actually splicing in a different protein into the original protein of interest. So we've got this chimera, this additional protein spliced into a GPCR here. And that can be to help stabilize it, to crystallize it, that sort of thing. So we always need to be keeping an eye out in the annotations for the PDBs for things like these. You get a lot of partial protein structures as well. And this is kind of a hazard of, sometimes proteins are, an ordered region, a flexible region, and an ordered region. And you'll only get the structure of the ordered region and get the two ordered regions apart from each other without the flexible bit in between. Just because that can make the flexible bit in between can make it really hard to crystallize and really hard to look at with structural biology techniques. And then of course, there are a lot of times missing pieces. So again, flexible pieces are most likely to be missing. A lot of times glycosylations are not included, things like that. So now I'm going to jump over to the PDB and we'll start walking through. So I'm going to start with this really simple PDB entry, just to show some of the basics. So this is actually myoglobin, which was the very first PDB entry ever deposited. And I pulled it up because it is a really good example of what a PDB file actually is. So I'm going to go over here to display files and PDB format. And what this does is it opens up the actual text of the PDB file. And if you look inside here, there's a lot of information that is exactly the same as what's over here on the entry page. Title, experimental information. So all along the top as we scroll down, there's just a lot of information about how the experiment was done. And remember, this is primarily so a structural biologist can communicate to another structural biologist all the details about their experiment and how they created their model. Now, if we keep scrolling, we eventually get into the meat of the protein data bank file. So what we see here is actually a list of every atom in the protein and a set of XYZ coordinates. So this is what actually gives you the 3D information so that a molecular viewer can then pull it up and create a visualization. So for example, atom 1, we can see it's a nitrogen. It's in the middle of a valine, which is a type of amino acid. It's in chain A, so that's what this A means here. And these are its coordinates in XYZ space. And we get an entry for every single atom that there is. Now, if we were to go and look at this protein in 3D view, and I'm going to change the representation here from cartoon to space fill and change my color from rainbow to by element, just to show you guys this idea. So for example, this nitrogen that's pointing out, if I cover my mouse over it, I can see, okay, so this atom is from a lysine, which is number 63 in the primary sequence. It's in chain A. And its 3D space is being determined, or its location in 3D space is being determined by this information in the PDB file. So that's how those two things relate. And that's what PDB files actually are. All this experimental information and then a list of atoms and where they are. So we also had that chain annotation, which is really important, but this is a pretty simple and sort of old and outdated entry. So I'm going to actually switch to a PDB file I had to work on recently for a project, the one that was in the banner. So let's switch it over to a slightly more complicated PDB. So this is 4UN3, which is actually the structure of a Cas9 protein. And it's a good example to look at the overall structure of a PDB entry. So up here we've got our ID and we have our title. Sometimes this title might be the same as the paper title down in the literature panel. But here they've actually named it something useful and helpful. So we see it's the crystal structure of Cas9 bound to PAM-containing DNA target. And this highlights, I think, one of my first tips for when you're starting to look at a PDB entry. So say you've been given a PDB ID and you're diving into parse, what's included. Make sure you do some background research about your protein before you jump in, because there's going to be a lot of specialized information within the PDB file. So this PAM-containing DNA target, if you've done some background research, you know, okay, so the PAM sequence is the part of the DNA that tells Cas9 to bind to this DNA. So Cas9 doesn't engage with DNA, it only engages with DNA that has this specific sequence that says, hey, look here. Okay, so titles, sometimes that's helpful, sometimes it's just the same as the paper and it can be a little less helpful. Another interesting thing here up at the top is this image, and you'll see it's labeled biological assembly, and we've got an arrow to click over here. So if I click over, there's also an image labeled asymmetric unit. And in this case of this protein, they are the same thing. But with the, in some cases, this will be different. The biological assembly is what the authors think is the biologically relevant occurrence of the molecule. The asymmetric unit is the smallest repeating unit within the protein crystal in X-ray crystallography studies. So to actually take a look at what I mean by that, let's go back over to our 3D viewer, which can be, again, really handy if we want to analyze something. So here we're looking at bioassembly 1, if I look over to the right here. But if I click down, I can select asymmetric unit, and that's still the same thing. And I can also click unit cell. And now what I can see here is actually how these proteins were packed into their crystal structure for X-ray crystallography study. And this can be important if you're looking at a protein and it has some funny bit kind of like sticking off the side, and you're comparing it maybe to like other visualizations or other PDBs and they don't have a funny bit sticking off the side. It's worth checking here to see if that might just be an artifact of these proteins sticking together. Is that a place where they're overlapping, and are they maybe kind of stabilizing each other in an abnormal conformation? Okay, so that's biological assembly and the asymmetric unit. In this case, it's the same. Under the download files tab, you do have the option to download the biological assembly, whereas the PDB format will be the asymmetric unit. Again, in this case, that's fine, but we'll look at an example later where these things are actually different. Now, as we scroll down, we see this, red-blue graph here. This is, again, scientists talking to scientists, basically evaluating how well the model was fit to its density map. And if we look over here, we have another handy little panel, the macromolecule content. It has our total structural weight in Daltons. So just shy of 200,000 Daltons or 200 kilodaltons. As you start to work with more molecules, like that will kind of give you an instinctive or an instant sense of like how large this molecule is, as will this image, because you can see how large the alpha helices are relative to the whole thing. But this is another handy place to just kind of look, see how big it is. It tells you how many atoms it has, how many residues. So in which case here, when they say residues, they mean both the amino acids and also things like water that might be in the structure. And then we see unique protein chains, 1, and unique nucleic acid chains, 3. And I'll come back to that in a minute. So we also have a really handy panel here, the literature panel. So this is the paper that is associated with this structure. And for some structures, you will see they do not have a publication associated. So they might be deposited before a manuscript is published, or a lot of times I'll run into structures that are deposited, but there's actually no publication ever linked to it and no associated publication. I do try to avoid those because when you see a paper here, it means that this structure, or at least the associated paper, went through peer review. So you're looking at like a legit structure, not something that somebody kind of threw together, but then nobody else ever double checked and then they just put on to the protein data bank. So I always like to use structures that have papers associated, again, to give me that verification this has been peer reviewed. And there are several helpful links here. So it links out to the PubMed entry for the associated article. And this can be really helpful because a lot of times in the introduction of these papers, they'll break down what the different pieces of the protein are, which you might need to know if you're kind of confused about, what domains they're talking about. They might have a nice intro here. And then there you can also see what other papers that they cite. So if you're doing background research to fill in the story around your protein, this can get you pointed in the right direction. And then we also see related structures. So a lot of times what happens, particularly now, before it might have been one structure, one paper. But a lot of times what happens is they have one paper where they're trying to put together the pieces of a story, and they'll have multiple structures that are involved in that. So there are actually 3 different structures in the PDP that are associated with this one paper. And also, if you're looking at this one, you're like, actually, this isn't the right structure, but it's close. One of these guys might be the right one. So let's say open for UN4. And here I can see that this is Cas9 bound to the PAM-containing DNA target, but then we also have mismatches at positions 1 through 2. So this is a variant of this first structure. And for what I needed, I just needed the kind of vanilla Cas9 bound to PAM-containing DNA target, just the standard, no mismatches needed. So I'll close that, but so sometimes this can be handy. All right, if we scroll down further on the page, we have the macromolecules panel, which is my absolute favorite panel of all time. I'll come back to that in a second. Below that, we have the less exciting small molecules panel, but this can also be handy. So here you'll find ligands listed. In this case, we just have a magnesium ion, not too exciting. But if you are, say, looking at a structure that has a drug bound or an important ligand, you'll actually see a 2D diagram of that. And that can be helpful if you're confirming, is this the drug that I'm looking for? Is this a closely shaped molecule to that? And you can look here to confirm that that's what you're looking for. All right, back up to my favorite panel. So the macromolecules panel. This has basically just about everything that you need to know about this protein structure. So first we've got the molecule title, and this is something that the authors have populated. So this can vary a little bit. and they have what chain that molecule is within the PDB file. So here we see it's chain B. It's about 1,300 or over 1,300 amino acids long. We can see what organism it comes from. And on top of that, we see this link to UniProt KB. So this link goes to the UniProt entry for this protein. And that will give us a lot of peripheral information about it. And it's linked to this through the primary sequence. So in the UniProt KB, if we open this up in a new tab, we get a lot of background information on the protein, its function, that sort of thing. We get its gene name. And we also get its sequence, which looks probably familiar compared to what I just showed you guys when we were talking about primary sequence, where we've got all the amino acids in order from N terminus down to C terminus. Now this PDB is mapped to that entry. So this gray bar is a representation of that sequence from N terminus to C terminus. And this blue bar says that chain B of 4UN3 maps to this extent of the primary sequence. And these little gray bits just indicate places where there are missing pieces. So they included these amino acids in their study, but maybe they were too flexible or something like that. They weren't able to actually see them. But it does look like we have nonetheless a fairly complete coverage of the primary sequence here. So that means the whole protein is pretty much in this PDB file, and it's in chain B. Now, you'll be like, okay, so that's chain B, where's chain A and all that? So if we look up here, this protein or this PDB has two different kinds of macromolecules, and it has the protein, but it also has nucleic acids. So if I click over here, I can see the nucleic acids listed out, and there you find this is where chain A is. Chain A is actually annotated as sgRNA, which again, having done background research, I know, okay, the single guide RNA. So this is the RNA that Cas9 uses as a template to compare to when it's checking DNA to see, should I cut this DNA? It will only cut the DNA if it matches this sgRNA. And there are a couple other chains listed here. So we've got target DNA strand is chain C, and non-target DNA strand is chain D. So let's actually go look at this PDB in 3D and correlate this information. So I'm going to go to 3D view. And I'm going to leave this in ribbon representation. I'm going to change the coloration from rainbow to by chain. And now we can see we have 4 different chains. We've got light blue, dark blue, red, and orange. And if I hover my mouse over, it'll tell me what atom I'm hovering over. In which case, this one is a lysine. It's number 1263 in its sequence, and it's in chain B. So that's what that B means after the 1263. And that lets me know, okay, so this light blue is chain B. So this is the protein Cas9. But if I hover over this dark blue, I can see the atom that I'm hovering over is in chain A. So that means that the dark blue is therefore the RNA that we were looking at back in our macromolecules panel. And then if we hover over this red strand, we see that it's chain D. So that's one of our DNA chains. And this orange strand is chain C. So that's the other one of our DNA chains. So this is something I do all the time, is just go look at the macromolecules panel, see what's A, what's B, what's C, and then come and look over here to put that together in 3D space and be like, okay, so that that's what's actually included in this PDB entry. So there's also a couple more things in this macromolecules panel. So all of this information in green is being pulled from the UniProt database. So it has information on if there are different domains that have been identified. So in this case, we have the PAM interacting domain. So this is the part of the molecule that would bind, or would bind to the PAM sequence in the DNA. And now with any one of these, if you want to learn more about it, you can hover over it and you'll see my pointer actually turns into a little hand. And this is a glitch that's been happening in all browsers that I've tested. But if I zoom out and I do the same thing, hover over, say, this PAM interacting domain, and I look 10 feet up in the page, you'll see there's actually a correlating expanded tab that shows up in black. So let me scroll up and scroll on just so you can kind of blink it on here. So that little tab tells me more in detail about that item in the macromolecules panel. So if I wasn't able to read this within the little green bar, that would tell me more information. Or if I come down here to the UP sites and say I hover over this little blue lollipop, this tells me that this particular amino acid is the proton acceptor during the enzymatic activity of the protein, and that its number is number 840. So this can be a really great place to look if you need to know about specific sites within the protein, if you need to know, what's my active site, what gets phosphorylated, that sort of thing. So if I scroll over these lollipops, you can see they're just different annotations for known amino acids that interact with different things. We also have a little bit of annotation about the secondary structure down here. So in red, we have the alpha helices annotated. In gray, it's just as coil, so that's something that's not structured. And in yellow, we have beta strands. And you can see that, I realize my screen is really small right now, just so you can see these little black tabs popping up as I scroll over things. But as I scroll over that beta strand, it says this is a beta strand, and it encompasses amino acids 1156 to 1167. So that can be helpful to know if you're looking in particular detail at a protein. Okay, another really cool thing about the macromolecules panel, let me scroll or zoom back in. It has this little gray plus bottom, or plus button. Oh, and a quick note, sometimes there'll be multiple proteins in the macromolecules panel, and then they'll only have the protein feature view open for one of them. And if it's not the one that you need, or you need to open it, you just need to come to this little protein feature view toggle. So I have a little screenshot of that in the handout with like a little pink star next to it. Just make sure you can find it. So if it's off and you need it, you can just turn it on here. All right, so another cool thing about the macromolecules panel, we have this little gray plus sign here. And if I click on that, it allows me, or it brings me to a page where this is every PDB entry that matches that primary sequence. So if I scroll down and I get another protein feature view panel here, I'm going to click that gray plus again. What you can see here is that these are every PDB that is on file that's mapped to this Cas9 sequence, or to this particular Cas9. And you can see some are, longer or shorter than others. So for whatever reason, these guys at the bottom, these short blue bars, are only a very short piece of the Cas9 protein. But for the most part, all of these other PDBs include pretty much the whole protein. some are missing pieces that they can't resolve. So these little gray bars. And some have more engineered mutations. So that's what these little tags that say E, those are engineered mutations. And if you're not sure what something is, you can again just hover over it and get annotation. I won't zoom out again, but I promise you as I'm hovering over this, there's a little black tab 10 feet up on the page somewhere that's telling me what it is. So this can be one way if you need to find the same protein but a different conformation. This can be one place to look. In this particular case, knew that I needed Cas9 down to just its RNA because the PDB that I started with for UN3, that is Cas9. It has its guide RNA, but it's also bound to its target DNA. And in the illustration I eventually did, I actually only wanted to show it just starting to interact with the DNA, but not actually having split it open yet to compare it to its guide RNA. So one way we can look for that structure is to, click on all of these and open the entry pages. So if I click on one, I can go to the structure summary page, and that would bring me back to my PDB entry. But there's another way to look at all the proteins mapped to this one sequence. And that is up here. So you see the number 34, and that tells you how many PDBs exist that are mapped to this primary sequence. And it has a little search icon next to it. So I'm going to click on that. And what that does is it brings up all of those same PDBs, that list we were looking at as a bunch of blue bars. It now brings it up as a standard PDB search where I can look at a little bit more information about the protein and about the PDB entry to try to figure out, okay, which one is, just Cas9 and the RNA. So I'm going to filter down a bit. So this little guy here, that's got to be that, little structure that's just a small section of it. So I know I'm going to go over here to polymer type, and I'm going to click mix because I know what I'm looking for is a mix of protein and nucleic acid. So click mixed, and that filters out the ones that are just protein. Okay, so now I'm still seeing a lot of structures. Fortunately, now that I've clicked on that polymer type, it gives me two more options. I can pick one that's both protein, DNA, and RNA, or there are 6 options that are just protein and RNA. That's what I need. So I'm going to click on that, and now I'm filtered down to 6 entries. All right, so now this is where it gets a little trickier. We have to figure out which one of these is, say, the canonical standard, just Cas9 with an RNA. And I can kind of already guess by structure or by title that these top entries are a little more involved than what I actually need. So these are, we've got Cas9 plus a single guide RNA, but also it sounds like some other proteins. So ACR2A2 or something like that. So those probably aren't what I need. Another way to kind of look for the canonical version of a protein is actually to sort these by release date instead of newest to oldest, oldest to newest. And that will get us toward most likely the more simple versions of the proteins are published first. So the structural biologists are most likely to solve, okay, just Cas9 and an RNA. And then later they'll do experiments building on that where it's like, okay, when it's bound to this thing or bound to that thing. Not always the case, but a lot of times the case. So you kind of have to use your context clues here to pick the right one. Okay, these structures are looking a little better, and there are a couple more context clues that tell me that these guys that I now have at the top are probably right. I can see one of the authors has the last name Doudna, as in Jennifer Doudna, and she is, the pioneering CRISPR scientist. So see her name on the publication. Okay, so this is probably the correct structure of CRISPR. I can also see that this was published in Science. So again, a lot of times the first time that a protein structure is resolved, it will get published in kind of one of those big big name journals like Science, Nature, which you're kind of seeing here. And then later, when they start to do more iterations on that, you'll start to see it's slightly less likely that you'll see them in like Cell and Nature. Again, this is more art than science. These are just some of the context clues that I take into account when I'm sorting through and picking out my PDBs. So now looking at these context clues, I'm also, of course, going to look at the title. So this tells me this is catalytically active Cas9 in complex with a single guide RNA. And that's exactly what I need. We have some extra details at 2.9 angstrom resolution. That's nice. Crystal structure, that's nice. But what I'm really looking at here is, okay, we've got Cas9 in complex with a single guide RNA. And if I look down at this one, it's nuclease inactive Cas9 in complex with a single guide RNA. And these are both associated with the same structure. I probably want the catalytically active one, so I'll click on that one. And if I look down, I can come down to my macromolecules entry and it says, okay, here's Cas9. And I know I'm going a little long, so 3 more minutes and then I'll open it up to questions. If I come down to my macromolecules panel, we've got CRISPR-associated endonuclease Cas9. That's the right name, named a little differently than the last entry. That's fine. I see it's A and C. Well, this is the thing where our biological assembly and our asymmetric unit are actually 2 different things. So let's go up and look at our picture. So we have a picture of the biological assembly, and that looks reasonable. It looks like we've got RNA and 1 protein. But if we click on the arrow, we have a second biological assembly, and then we have an asymmetric unit, which is actually 2 of the proteins together. And this is just how the crystal happened to form. The biological assemblies tell me that the authors say, okay, these biological assemblies, that's the biologically relevant unit. So if you're going to visualize this protein, you want to use a biological assembly. And because the asymmetric unit was 2 of them, they ended up in the course of their experiment solving 2 slightly different shaped models for this protein. It doesn't really matter which one that you use, unless they're dramatically different and one has like a funny shape sticking out of the side that looks a little weird. You might not want to use that one. But either way, when you go and you use this PDB, you want to use one of the biological assemblies, probably biological assembly 1. But if you download and open just the PDB, you're going to get the asymmetric unit. So you'll have 2 copies when you only need 1. Yeah, so I think that's everything I wanted to say about that. So I will open it up to questions.
Thanks, Veronica. That was so informative. We have just a few questions coming in, so everyone else, feel free to type in your questions. I'm going to start at the top. So we have a question from Isun Wu. It says, thanks for this great presentation. One question I have is how to read the PDB validation. If there are multiple of the same structure, how to pick a better or more suitable one based on the percentile ranks?
Oh, okay. That is a great question. I actually never do that. But let me say, there's kind of a quicker way. Usually I don't have to decide. Usually I don't have to look here. I ignore over here. Sometimes what I'll do is if I find a bunch of entries that are otherwise completely equal, I'll go down to my handy macromolecules panel, I hit my Great Plus. And if I scroll down here and hit my little Great Plus, hit my Great Plus again, one of the things I can do up here is color by resolution. So I can, see which one's a higher resolution, for example. So maybe, an older structure was solved at a relatively low resolution, and there's a newer one that's higher. As you can see, most of these are all kind of, as far as X-ray crystallographers are concerned, not great resolution. That's why they're orange and red, except these 2 little ones. They're blue, they're okay. So that's one way. So if I'm trying to decide between 2 otherwise equal structures, I might look at that. Another thing else. Oh, go ahead.
Colors?
I think,
Know what's good and what's not good.
yeah, I know there's a key somewhere. If you scroll down to the bottom, usually they have annotation. See if they have, I click on this. I feel like it should be overlaid with it. If I click on the entry, like this red one, and go to structure summary, it should say the resolution, yeah, 5.2 angstroms. That's pretty bad. Well, that's good for cryo-EM, not that great for a structure. And cryo-EM is getting better, so I'm kind of surprised to see 5.2 here. Anyway, so 5.2, that's kind of high. So that's why that's red. But if we look at one of these blue guys and we go to the structure summary, you can see that the resolution here is 1.9 angstroms. So resolution is like golf, the lower the number, the better. So 1 to 2, that's really, really good. Like 2 to 4, decent. 4 and above, and I should say that's not like a really hard and fast delineation. That's just kind of my field, having worked with structural biologists, and like the numbers they're usually aiming for, and they want to get at the very least under 4 angstrom resolution. And another way that you can kind of decide which one's better is, look here and be like, do you have the complete protein? Some of these guys have resolved all of the amino acids, but then other ones have kind of big missing chunks, indicated by these gray bars. So that might be another way to be like, okay, I don't want to use this one. I want to use this one that's more complete, or has fewer engineered mutations, that sort of thing.
Yeah, that's a great lead into the next question. So this is from Stephanie Jones. So in the case where the entire protein hasn't been assembled in one file together, or you have to piece a couple different files together, any tips on how to arrange the pieces together? Or another example would be a receptor ligand pair that haven't been put into the same file together, how to arrange them together in the correct conformations.
The challenges. Yeah, so let me first start with the when something's all different pieces, which I was actually had an example, but then ran out of time. So let's look at this. This is a receptor tyrosine kinase. And let's say you've been given this PDB that is the intracellular domain. But it's just the intracellular domain. If I go down to the macromolecules panel, I can click down here, hit my little plus button again. And here I can see all the different pieces that, oh, I don't know where it went. That's odd. Sorry about that. Okay, and we're back. So here I can see the different segments of the protein. And remember, everything is ordered from N-terminus to C-terminus. So if you are going to take, this extracellular portion and stick it together with a transmembrane portion and stick it together with the intracellular portion, and by the way I know that these are these things, you can see this little annotation up here that says extracellular, helical, cytoplasmic. So that's what I'm looking at. So that's how I know this is the extracellular piece, this is the transmembrane piece, this is the cytoplasmic piece. You want to make sure that, if your N-terminal is over here and your C-terminal is over here, that the C-terminal or the C-terminus of the extracellular piece is pointed toward the N-terminus of your transmembrane piece. And the C-terminus of your transmembrane piece is pointed toward the N-terminus of your your intracellular domain. Other than that, they probably aren't solved together because they're maybe kind of flexible, that sort of thing. You kind of just line them up, and that's kind of the best guess of what the whole structure looks like in this case, where there isn't anything that is all of it all together. As far as the other question is, how do you fit a ligand with a receptor? And if that structure hasn't been determined yet, hopefully if you're being asked to do that, they already know at least one similarly shaped ligand. that has been seen bound to that receptor. So you at least know what binding site it goes into. So a lot of times what you can do is you can compare a known ligand versus an unknown ligand and then be like, okay, so they're like similarly shaped in this region, and kind of try to line up those two similarly shaped regions. I don't have an example for that, but yeah, and that's where that little viewer, let me just open any of these guys. So that is where this small molecules panel comes in handy, because you can see the 2D structure of the ligand. And it might be really close, but not quite exactly what you need. And that will tell you, okay, if this protein that is like, or this ligand that's really close to the one that I need to fit is in, the main binding site, then the one that I need to fit probably also fits in the main binding site in a very similar orientation.
Thanks.
Okay, I'm gonna cut it off there. There's a few more questions, but as I recall from the from actually watching this thing a few weeks ago, the remaining questions are not quite as interesting. I did think a couple, like if you have several different fragments of a protein, but nobody has actually solved the entire thing all in one piece, how would you know how to line up the extracellular domain, the transmembrane domain, and the intracellular domain? I think that's pretty interesting stuff. But we are just about out of time for today. I realize that's a lot of material all at once, but fortunately it is recorded. And so this will be up with the cloud recordings, the Zoom cloud recordings in my courses. It may take a little while for it to process, but it will be up there. So let me stop sharing my screen here. And I'm going to unmute everybody. Those of you who are still awake and still with me. Anybody have any last question or any questions before we go? Well, there were a few that showed up in the chat in case anybody wanted to take a look at those. And anybody else have any questions they wanted to shout out?
I have a question.
Yeah.
When I was dealing with the acetylcholine receptor, I find I cannot using a surface model. Is that because that molecule is too large?
Oh, sometimes. Like, are you talking about in Chimera?
Yeah, in Chimera.
Yeah, sometimes if the molecule is very large, you'll get an error when you try to add a surface to it. Sometimes it's a limitation of the memory of your computer.
Oh.
Like I've done a lot of acetylcholine receptors. I've illustrated quite a few, and I've never had trouble putting a surface on it. So I don't know if it's if it could have something to do with the computer, or maybe just that particular model just has a little glitch in it. If you run into a problem like that, you could actually, you could try to find a different model of the acetylcholine receptor, like that OPM database, the Orientation of Proteins and Membranes, had, I don't know, it had like 8 different G protein coupled versions of the acetylcholine receptor. It had 8 different versions of the ligand-gated ion channel. So it, there's a lot of them out there. You could just try possibly a different one also.
Actually, I can try like a different version maybe.
And if worse comes to worse, you could always possibly try a different program like VMD or Pymol and see if that'll let you put the surface on and export it. Especially if you're gonna then take that surface and try to bring it into a 3D program to actually render it.
I had a similar problem when I was working with I'm trying to look at the CFTR protein and throw a surface onto it, and it gave me another.
Have you pet the puppy? Do you like her?
I do like the puppy.
I'm sorry, you ran into the same problem, Austin, that it
Yeah.
just did it give you an error message? And,
Yeah, it's for whatever reason. Let me see here. Sure. Yeah, it says the surface calculation frequently fails for large multi-change structures.
Huh.
Um,
yeah, I'm not sure what to say about that. For CFTR, I don't know if you're going to find a lot of other ones out there that you could try. I mean, they're all going to be just as big because, you know, CFTR is CFTR. There's not, you know, they're all going to be big files, I would think. But I am wondering if it's a memory issue, just because, you know, that's CFTR is another one that I've rendered before, and don't recall having problems with it. So,
gosh, I hope 16 gigabytes of memory would be sufficient, but
16 gigs of RAM, yeah, you'd think it should be.
I had maybe like a similar issue when I was trying to put a surface on the transcription release protein, and
Yeah.
it gave me like, I don't remember exactly what the error was, but it sounded like something similar, but it put the surface on it anyway, and it didn't look like anything was missing.
Yeah, I've had that happen many times where it gives you the error, but then it draws it anyway. So,
Yeah, so I ignored it, but I don't know why
But
it would do that.
in your case, Austin, and same thing with Kiwan, it gave you an error and it just didn't draw it.
yeah,
Yeah. Mm.
it puts it on the ligands. When I select chain A for the protein, it actually selects everything. But you can also select the ligands separately with the structure selection, which is strange.
Yeah.
I've tried like removing those and then adding a surface, and it also gives the same error message.
Huh.
Mine is like all the small ones, the small molecules all works. It's just like when it came to the big ones, it just doesn't show up.
Oh, that's okay. Yeah, I'm not sure what's going on with that. That could be a problem because our next fairly large exercise does involve doing a surface representation of a protein. So, I mean, we're going to redraw it in Illustrator, but you still have to generate the surface to get the reference. So, I mean, it may require using one of the other programs and see if VMD or PyMol will do it, or possibly searching for a different version of the molecule. But you know, like I suggested to Kiwan, but you know, they're all going to be big files. So I haven't run across that before. I'll have to think about that. But since I haven't run into it, I don't have any workarounds. All right, well, we are officially out of time here. So I'm going to stop the recording and let you go. But since that is recorded, you know, that'll be something that you can refer back to as a reference for the future. Probably way more than you wanted to know about the protein data bank, but as you can tell from Veronica's webinar, it's an extremely powerful thing once you really get to know it and get familiar with it.
Jim, where do we find these recordings? Are they on my courses or?
Yes. When you go into my courses and you select this particular class, at the top of any class that you're enrolled in, you know, you go to that class, there'll be a bunch of tabs at the top, and there's actually a Zoom tab at the top of each class. And when you go in there, there should be a link for cloud recordings.
Oh, okay. Thank you.
Yep. So the one from Monday should already be up there, at least, you know, when I realized halfway through class to record the second half of it. But today's, the whole thing should be up there.
Okay, thank you.
Yep. Okay, don't see anything else popping up in chat. Like I said, we are right out of time, so I'm going to go ahead and stop the recording.